All right, let's jump into our very last chapter of organic chemistry. It's all about amino acids, peptides, and proteins. And a lot of you that have taken basic biology courses have actually talked about this in a little bit of detail, but we're going to dive into the chemistry. And if you think about an amino acid, it obviously has two different functional groups. It's got an amine functional group on one end and a carboxylic acid functional group on the other end. In addition, there are a variety of different R group chains that come off to the side, but there's a whole bunch of these amino acids that exist in nature. If you go online, you can find a variety of charts that contain these amino acids. Here's a chart that I really like from compoundinterest.com, and you can see the R groups vary off of each of these. So for example, you could have a methyl group coming off the end. You could have nothing coming off, or you could have some sort of sec-butyl group coming off. There's a variety of different R groups on here. The nature of these R groups can impact the behavior, or I should say physical properties, of each of these amino acids. So if you look over here, you'll see that they've kind of classified everything into these basic groups in color coding. So we've got aliphatic amino acids. These are circled in red. That means that their side chain contains alkanes. The green ones right here are aromatic. Obviously, they have aromatic side chains. We've got acidic ones, basic ones, ones with hydroxy groups or alcohols, sulfur-containing ones, ones containing other amides within them. And we've got both non-essential ones and essential ones. So for humans, the essential ones are usually included in most of the food that we eat. However, if you have any friends that are vegetarian or vegan, they may actually have to supplement in some of these essential amino acids to ensure that they're getting a complete and balanced diet. It just depends a lot on the food that you eat. In addition, when you look at this, they've got a code over here. One of the things we do is instead of writing out the full name, we just abbreviate it. So for example, alanine would be abbreviated ALA, or the single letter code would just be A. And if you remember from biology, these are all coded for in our DNA using these codons. We're really not going to get into that in this course, but it's covered fairly extensively in basic biology. So all of this is basically stored genetically, and then the organism can produce it using something like the ribosome. All right, the cool thing is each of these amino acids can be linked together either biologically or synthetically into a polypeptide, which is basically just a big polymer of amino acids. So let's go ahead and draw this backbone out, and then I'll add on my side chains here in a second. And I'm going to bracket this off kind of like we did in the peptide chapter, and this could repeat a certain number of times. This R group would come off here as a dash, over here it would be a wedge, over here it would be a dash, and it would just repeat over and over and over again. So this is just referred to as a polypeptide. Nature is really good at doing this fast. Chemists have struggled with this a little bit more, but we'll talk more about how we can do this synthetically in a lab. It's kind of interesting. All right, I did want to show you another way we can draw peptides, and we can draw them using Fischer reps, or representations. And we did this a fair amount using Fischer representations for carbohydrates. And if you recall, with carbohydrates, we oftentimes drew them kind of like this. And we would have an OH group coming off here and here. And we said that all natural carbohydrates have this lower OH group off to the right. That means that these are D sugars. So this is a carbohydrate. Let's compare this to our amino acid. And we can draw amino acids using Fischer representations, but this time we're going to have a carboxylic acid group on the top, and we're going to put the R group down at the tail. And when we do this, we still need our amine, but this time it's going to be off to the left, not off to the right. And so this time, because it's off to the left, we need to use a different designation. We can't call it a D amino acid because it's not off to the right, it's off to the left. So we call this an L amino acid. So L equals group to the left. And so nearly all natural amino acids are L amino acids, but I actually read an interesting article a few years ago talking about these toxic mushrooms that were killing people in China. They were accidentally eating them, not quite knowing what they were. And so they isolated the compounds that were toxic. And what they found out was that some fungi can actually make L amino acids, which are really, really bad for you. Your body doesn't know what to do with them, and it kind of mucks up your entire system. So it's better to avoid the D amino acids tend to be quite toxic, and they're found in a handful of plants and fungi. All right. The cool thing with amino acids is the basic chemistry that they do. No pun intended. But it can do a variety of acid-base chemistry. So let's say we've got this generic amino acid. The neat thing with it is we have two sites. Over here, we've got this site that's obviously acidic, right? It's a carboxylic acid group over here. But over here, we've got a basic site. So in this way, it can act as either an acid or a base, meaning it can do either. So it can do acid and base chemistry. And let's just take physiological pH, meaning the pH in your body. So under physiological pH, meaning the pH inside your body, the acid is going to lose its proton, right? So the carboxylic acid in your body would prefer to lose that proton and form the carboxylate salt, right? So over here at that physiological pH, it'll form a carboxylate. Let me zoom out a little bit. And down here, the amine, because it's basic, will get protonated at a physiological pH and therefore have a positive charge. So if you remember, this is called an ammonium group. The IUM suffix indicates that it's positively charged. So in this case, what we've formed is a Zwitter ion. And a Zwitter ion, if you remember from first term, is just a molecule that has both a positive and negative charge, but no net overall charge. All right, under low pH, meaning acidic conditions, the acid isn't going to get deprotonated, right? Because we're under acidic conditions. So that's going to stay put. And it's just going to remain as a carboxylic acid. However, the amine will get protonated and it will have a positive charge. So under low pH conditions, these amino acids tend to act as cations. Under high pH, you can imagine the opposite is true. So under high pH conditions, what we'll do is we'll deprotonate our carboxylic acid and our amine will stay neutral. So in this case, we're dealing with mostly anions. So depending on the pH that you're working with, you can either have your neutral species, which is a Zwitter ion, you can have your positive species, or you can have your negative species. So we can actually control a lot of the physical properties by adjusting the pH. All right, if you look in your textbook, there's actually a pretty good table that talks about this in more detail. This is table 25.2, I believe in most editions. And this table lists the pKa values for the various amino acids. And it's a little bit confusing, but what it does is it lists the pKa values for the carboxylic acids, and the ammonium ions. And this is something that always threw me off when I was an undergrad, was I always thought a pKa was just looking at your neutral molecule, but what it's saying is, what is the pKa of this ammonium ion. How easy is it to remove that proton from the ammonium ion? Is it really hanging on to that proton, or would it rather dump it off on some other base? And in addition, it's saying, what's the pKa of the carboxylic acid group? What some students do, though, is they look at it and they say, well, isn't it the pKa of that? No, not exactly. The pKa of amines are so high that it's not even really worth listing because they're not really even experimentally that valid for most physiological studies. It's a lot easier to study the pKa values of these ammonium ions. So keep that in mind when you're looking at the table. All right, there's one last thing I wanted to cover, and this is called the isoelectric point. And the isoelectric point is the pH at which the Zwitter ionic form is at the highest concentration. So in the example above, I said that under physiological conditions, we form the Zwitter ion. But it's not exclusively formed, right? We're always dealing with equilibria. So it's saying, what's the pH at which the Zwitter ionic form is at the highest concentration? Oops. And this can be experimentally studied. However, it's kind of nice if you already know the pKa values from the table I showed you above. And what you can do is you can calculate this isoelectric point, which is abbreviated Pi. And you can calculate it by first taking the pKa of your carboxylic acid for a specific amino acid, add that to the pKa of your ammonium ion, and divide this by 2. And this will essentially give you the pH for your isoelectric point, which is also just referred to as Pi. The useful thing with doing this is, like I said up here, each of these has very different physical properties. Something that's an anion is going to behave very differently when you're separating it from a cation, and that's going to be very different than separating it from a neutral molecule. So if we do something like chromatography, we can separate these out pretty easily. We can even use gel electrophoresis to separate these out based on their charge. So that was going to be my last point, is that we can purify these. And I'm just going to call these amino acids, AAs, and or polypeptides, using gel electrophoresis at a proper pH. So if you ever go on into a biochemistry lab and do some of this synthetic chemistry, you might actually do some of this gel electrophoresis, but I wanted to introduce the basics concept. All right, when we come back, what we're going to do is we're going to look at how to make amino acids synthetically. And that involves a lot of older chemistry, but interesting chemistry.